Today, my talk will be on semaporin 5B controlling spiral ganglion neuron branch refinement during development. Semaporins are known as axon guidance cues in the nervous system. They are also extracellular signaling proteins that are essential for the development and maintenance of many organs and tissues. Semaporin signaling occurs predominantly through plexin receptors and results in changes to the cytoskeletal and adhesive machinery that regulates cellular morphology. These plexin receptors are what the semaporins use to get into the cell. There are 7 different classes of semaporins, and we're going to be talking about semaporin 5B and the plexin receptors A1 and A3. Hair cells are organized into one row of inner hair cells and 3 rows of outer hair cells. Hair cells are mechanoreceptors responsible for detecting sound waves and releasing glutamate onto spiral ganglion neurons. Inner hair cells are innervated by type 1 spiral ganglion neurons, and type 2 spiral ganglion neurons innervate about 15 or more outer hair cells. And you can see that here, where there's one spiral ganglion neuron on one inner hair cell, and there's multiple of them on the outer hair cells. Semaporin 5B controls the branching behaviors of auditory neurons within the cochlea. It's also been shown to eliminate synaptic connections in hippocampal neurons in vitro. It's expressed in developing cochlear hair cells and signals to plexin A1 on SGNs. The signaling appears to limit type 1 SGN branch refinement as part as SGN limits type 1 SGN branch numbers as part of SGN refinement. So axons with a greater complexity of terminal branches have a longer branch depth value, whereas axons with a more limited complexity have smaller branch depth values. Semaporin 5B FC treatment did not cause the SGNs to retract as if they were under the influence of a strong chemorepellent, which is a similar experiment that we observed that would affect the semaporin 3A FC. Sema 5B treatment led to a reduced terminal branch number and branch depth, and it also led to an increased branch length. SGNs with fewer numbers of branches tend to have longer individual branches, and the addition of exogenous Sema 5B does not cause an overall SGN retraction response, but that Sema 5B may alter how the SGN branches are configured during the refinement process. Loss of Sema 5B also yielded SGNs with reduced branch lengths. The loss of Sema 5B does not cause significant hearing impairment. It is possible that Sema 5B signaling induces either a local branch retraction event or alternatively a local branch stabilization event. Sema 5B FC gain-of-function experiments led to fewer but longer terminal branches, whereas loss of Sema 5B led to more numerous but shorter terminal branches. One possibility is that the Sema 5B mediated retraction events subsequently led to the elongation of neighboring branches because the cytoskeletal or other cytosolic components are redistributed. Alternatively, Sema 5B may stabilize select branches while others retract in response to the nearby repulsive cues. It's also been shown that plexin A1 and Sema 5B are predicted to have the same pathway. And plexin A1 is showed in the mRNA of the entire cochlea, but it's only shown in the middle, expressed in the middle turn of the cochlea. Here are my works cited. Thank you.